Have you ever wondered how far we can push the boundaries of genetics? The realm of genetic manipulation has fascinated us, sparking conversations and driving research for decades. It's a topic that's as vast as it is intriguing, filled with possibilities that were once only the domain of science fiction. From the early days of selective breeding to the discovery of DNA's double helix structure in the mid-20th century, we've been steadily peeling back the layers of life's blueprint. The dawn of genetic engineering marked a significant turning point, allowing us to manipulate genes within organisms, opening up a world of potential applications. Fast forward to today, and the field of genetics has evolved beyond our wildest dreams, with advancements happening at an unprecedented pace. But nothing has quite shaken the world of genetics as much as a revolutionary tool that's become a game changer for scientists worldwide. This brings us to the revolutionary tool that is redefining the field of genetics: CRISPR. CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, wasn't born overnight. It's a tale of curiosity, serendipity, and relentless scientific exploration spanning 3 decades. The saga kicked off in the late 1980s when Japanese scientists were examining a peculiar pattern in the DNA of the E. coli bacteria. They stumbled upon repeating palindromic sequences, broken up by spacer DNA from viruses that had previously attacked the bacteria. This was an intriguing puzzle, but its significance remained a mystery for years. Fast forward to the early 2000s, when the scientific community started connecting the dots. They realized that these repeating sequences, now known as CRISPR, were not just peculiar patterns. They were part of a bacterial immune system. When a virus attacks a bacterium, the bacterium snips a piece of the virus's DNA and tucks it into its own DNA, in between the repeating sequences. This serves as a mugshot of the virus, helping the bacterium recognize and fend off future attacks from the same virus. And then came the game changer. In 2012, Jennifer Doudna and Emmanuel Charpentier demonstrated that they could hijack this bacterial immune system and turn it into a tool for editing the DNA of any organism. They showed that an enzyme called Cas9, guided by a piece of RNA matching the DNA sequence to be edited, could act as a pair of molecular scissors, cutting the DNA at a precise location. This opened the door to fixing genetic mistakes, manipulating genes for research, and even creating genetically modified organisms. This discovery was like unearthing a hidden treasure. It was as if nature had been quietly perfecting this elegant system of genetic modification, and all we had to do was discover it, understand it, and harness it. This marked the birth of a new era in genetic engineering, but how exactly does CRISPR work? Imagine having a pair of scissors that could cut and paste DNA sequences. This isn't science fiction, but a reality made possible by a revolutionary tool known as CRISPR. Think of CRISPR as a pair of molecular scissors that can precisely edit the DNA of living organisms. At the heart of this system is a protein called Cas9. Cas9 acts as the blade for our genetic scissors, snipping DNA at specific locations. But how does it know where to cut? That's where guide RNAs come into play. These molecules are like GPS for our Cas9 protein, guiding it to the exact sequence of DNA that needs editing. Once the Cas9 protein reaches its destination, it makes a precise cut. Scientists can then add, delete, or replace DNA sequences, effectively rewriting the genetic code. Now that we have an understanding of how CRISPR works, let's delve into the real-world applications of this tool. CRISPR has brought about a paradigm shift in multiple fields. This revolutionary technology is paving the way for groundbreaking advancements, reshaping our world in remarkable ways. In medicine, for example, CRISPR is spearheading the fight against genetic disorders. Imagine being able to correct the genetic defects that cause diseases like cystic fibrosis or sickle cell anemia. With CRISPR, this isn't just a dream, it's becoming a reality. Scientists are already using it to treat certain types of blindness and cancer, and the possibilities for future treatments are virtually limitless. But the applications of CRISPR extend far beyond the medical field. In agriculture, for instance, it's being used to create crops that are more resilient, nutritious, and sustainable. It's enabling farmers to grow food that's resistant to pests, diseases, and harsh environmental conditions, helping to feed our ever-growing global population. And let's not forget about the energy sector. CRISPR is playing a key role in the development of biofuels, providing a cleaner, more sustainable alternative to fossil fuels. By modifying the DNA of algae, scientists are improving its ability to convert sunlight into energy, offering a promising solution to our planet's energy crisis. Even more astonishing is the potential of CRISPR to bring extinct species back to life. Picture a world where the woolly mammoth roams the earth once again, all thanks to CRISPR technology. While this may sound like science fiction, it's an idea that scientists are seriously exploring. These are just some of the ways that CRISPR is transforming our world. It's a tool of extraordinary power and versatility, one that's enabling us to rewrite life's blueprint and shape our own evolutionary destiny. Yet, as with any powerful tool, CRISPR also raises important ethical questions. As we continue to unlock its potential, we must also grapple with the profound moral implications of manipulating the building blocks of life. The power to edit life's blueprint is not without its controversies. As we step into this new era of genetic editing, we must grapple with the ethical implications of CRISPR. Its potential for misuse is palpable, stirring conversations about the need for stringent regulations. Moreover, the possibility of designer babies sparks debates on the boundaries of human manipulation. As we stand on the brink of a genetic revolution, it's crucial to navigate these ethical waters with care. The future of genetics is in our hands. As we look ahead, we see a world where CRISPR technology could eradicate genetic diseases, revolutionize agriculture, and even bring extinct species back to life. Yet with great power comes great responsibility. We must use this genetic tool wisely, ensuring we respect the natural world and maintain ethical boundaries. The CRISPR revolution has only just begun. The question is, how will we choose to write the story of our genetic future?